Hello, today is Friday, May 23rd, 2020. My name is Ricardo Sandoval, and this video is for week 2 of Biology 225, Microbiology at Los Angeles Pacific University for the summer 1 term. This is lab number 3, happening in week 2. So this laboratory experiment assignment is titled Bacterial Enumeration, Dilution, and Plate Count. So basically what I'm going to do is we're going to use the bacteria formed in yeast, which is S. cerevisiae. And S. cerevisiae stands for Saccharomyces cerevisiae. So basically what I'm going to do here is we're measuring what's called CFU per milliliter, and CFU stands for colony forming unit. Basically, in layman's terms, what CFU means, it's the amounts of viable bacteria in a sample. So to show you, I'll show you this applies here in a minute, but basically we're going to start with a master sample, if you will, of yeast with some water. And that's going to go into another 6 test tubes, and then each of those samples will have a small amount put into 6 petri dishes that have nutrient agar in them, and then we'll let them grow and see basically how many colony forming units or how much viable bacteria is in each. Basically, for a sample to be considered viable, we need to see or count 30 to 300 colony forming units, or CFUs. So with that being said, I'm going to go ahead and switch the camera angle to show you the supplies I'll be using for this experiment. It's just a little sign I made with the information so my name, the dates, the lab, the class, and the school. Here I have 6 petri dishes with agar, labeled 1, 2, 3, 4, 5, and 6. I have 6 test tubes here, so the first 3 at the front, this is 1, 2, 3, at the back, 4, 5, and 6. This is my yeast. So 1/4 teaspoon of yeast to 2 tablespoons of warm water that has bloomed. I have a pipette here that has some alcohol in it to sterilize, and then some water. I do understand that the experiment did call for distilled water, but given the constraints of obtaining items quickly and safely, and given the current COVID-19 pandemic, I was unable to secure distilled water for this experiment, which I hope won't taint the results. I also have gloves and my safety goggles, and I also have, of course, a mask. And then my work surface I've already sanitized with a 10% bleach solution. Okay, so with that being said, I will go ahead and start the experiment. For the most part, you'll just see my hands and you'll hear my voice. Okay, so the first thing I'm going to do here is I'm going to put 2.25 or 2 1/4 mils of water into each test tube here. So I have my pipette that's been sterilized. I'm just going to discard the alcohol in a trash can that I have here. I'll make sure it's nice and dry. Okay, so 2.25 mils in each of the 6 test tubes. So that's 1 mil, 2, 1/4. 1, 2, 1/4. 1, 2, 1/4. And test test tube number 4. 1, 2, and a fourth. Number 5, 1, 2, and a fourth. And then lastly, test tube number 6. 1 mil, 2, and a fourth. Okay, and then like I said earlier, I'm going to use what's called the master sample. So I'm going to pipette or add 1/4 milliliters of my yeast, which I have here, in the test tube 1. So just gonna swish this around, make sure everything's nice and mixed. Okay. So there's my 1/4 ml of yeast. And then what I'm gonna do now is I'm just gonna draw this up and push it back in several times just to make sure it's a nice homogeneous mixture. Okay. So now for the remaining 5 test tubes, I'm gonna pipette 1/4 ml of this into the remainder. So there's 1/4, and again, draw up and down. And as I go forward, the turbidity or cloudiness in each of these tubes is going to get lighter and lighter. Since I'm not adding yeast directly into the tubes, I'm just pulling from the master sample. That's why we're basically doing dilutions, so that each one is a bit weaker. And then from there we'll see our CFUs, or colony forming units. Okay, that's for test tube 4. Well, let's mix that up. And as you can see, at this point, I mean, it just looks like plain water. But there is a minor amount of yeast in there. Okay. This is number 5. Okay, and then lastly, number 6. Okay. And by far, this last test tube, number 6, is the lightest of them all. All right, so I'm going to go ahead and place my pipettes back into the alcohol. Okay, so now that I have my dilutions, 6 dilutions for these 6 agar plates that you see here, what I'm going to do now is I'm going to pipette 4 drops from each test tube. So for test tube 1, into dish 1, 2, 3, 4, 5, and 6. There's a fly in here for some reason, so excuse me. This California summer is just kind of bringing things about. Okay, so I'm just going to remove the lid here. This is test tube number 1, the cloudiest of all of them, and just 4 drops is all it's going to go in. So 1, 2, 3, and 4, and that's it. So now what I'm going to do, I'm going to close the lid, swirl it around, just to really distribute it amongst the agar, and set it aside. Okay. Test tube number 2. Let's draw up a small amount. And again, just 4 drops. 1, 2, 3, and 4. Lid goes on. Swirl. I'll set aside. This is number 3. One, 2, 3, 4 drops. Lid on. Swirl. Okay. Number 4. Just going to drop a small amount. 4 drops. 1, 2, 3, and 4. Lid on. And swirl. Okay. Number 5. Four drops. 1, 2, 3, 4. Lid on. Swirl. And then lastly, number 6. 4 drops. 1, 2, 3, and 4. Okay. So now I have my 6 samples, and again, lid on, swirl. Okay. So that's 6, 5, 4, 3, 2, and 1. Okay, so now these are going to sit in the closet of my office, my incubation location, for 12 hours in this direction. Then I'll flip them and let them sit for a total of 48 hours. And then we'll reconvene, if you will, to see how many colony forming units have completely developed. Thank you.